In the first video, I have already said the steps of the formation of the male gamete. Its first topic was that the development that leads to the formation of male gamete. Male gamete means microspores. The pollen or the pollens or the male gametes, that is the microspores, and the pollens are forming inside the anther, that is the sporangium. So the first is development of the anther, means microsporangium. This microsporangium is eusporangiate type, means a group of initial cells are first produced. Group of initial cells are first produced. Here initial cells are first produced. That type of development is known as sporangiate type. Some of the hypodermal cells differentiate to produce archisporial cells. Archisporial. These archisporial cells divide and to form outer parietal cells and inner sporogenous cells. Then the outer parietal cells produce one epidermis at outside. Below epidermis layer, another layer is there, that is the endothecium. This one is the endothecium. This one, endothecium layer. What is the role of this endothecium? Endothecium helps in the dehiscence. Dehiscence means splitting of anther. It splits anther and then anther bursts. So outer epidermis, just below epidermis, this endothecium helps in the dehiscence. Below endothecium there are 3 to 5 layers. They are called middle layers. Below middle layers another layer is there. This dotted structure of this green colored structure I have drawn over here. That is the tapetum. Tapetum. What is the role of tapetum? Tapetum is a nutritive layer. It provides nourishment to the microspore mother cells. Then below it there are microspore mother cells. So this is the anther lobes, 4 anther lobes. This is called microsporangium. One microsporangium I have drawn over here to show you that outer layer is epidermis. There below it different layers are there, and inside this tapetum. More zoom view of one microsporangium is this. So this is all about the development of anther, means microsporangium. First step was the development of the microsporangium, means the anther. Now the second step is the formation of the pollen grains, means microspores. Okay, and then look at the board. Question comes, what is microsporogenesis? So the answer will be the formation of microspore, means pollen grain is called microsporogenesis. Here the sporangial cells, I have said the sporangial cell was inside. Sporangial cells divide and produce pollen mother cell. Instead of pollen mother cell, you can write down microspore mother cells also. So sporangial cells divide and produce pollen mother cell or microspore mother cells. Now each cell divide meiotically. Okay, reductional division. They divide meiotically and produce 4 haploid cells, 4 haploid microspores. Now I will discuss structure of pollen grains, means microspore. This microspore or the pollen grain is uninucleated, means only one nucleus is present. Unicellular, only one cell. And this haploid N, chromosome number is N. And this is the first cell of the male gametophyte. It is surrounded by 2 layers, outside outer exine and inside intine. Outside exine is thick and intine is thin. This exine, there are some points where there is no exine. There is a germ pore, or the points through which the pollen tube emerges, that are known as germ pores. normally in dicot plant there are 3 germ pores, and in monocot plant there is 1 germ pore. Now look at the board to see the structure of microspore. Structure of microspore, it is unicellular, 1 cell, uninucleate, 1 nucleus, N chromosome number N, and it's a spherical structure. The first cell of male gametophyte, it is having 2 layers, outer exine thick, inner intine thin. Sporopollenin. Sporopollenin, this outgrowth contains some substances which is resistant and fatty substances. That substance, that sporopollenin, this substance cannot degrade by the digestive enzymes. It is resistant to alkali and acids. So that is very resistant substance, sporopollenin. Question comes from this chapter, what is sporopollenin? Okay, now look at the board. Sporopollenin. Next, germ pore. Germ pore are the points where from the pollen tube emerges. See, some gap-like structures are there. Here 3 I have dot. These are the germ pores. In dicot it is 3. In monocot the number of germ pore is 1. So that is the structure of microspore.